Thank you very much. I just wanted to start out by saying this is my first time attending Venox, and I want to say thank you to everyone. It's been a really incredible conference to attend, and everyone's been very friendly to someone like myself who is new into venom research. I'm going to continue to switch up what we're talking about a little bit today away from the snakes and introduce you a little bit to an understudied area of stony coral venoms, and specifically their impacts that climate change and environmental stresses may be applying to them. So to start out with a little bit of background, since I know there's probably very few, if any, people here who study coral, coral are in fact an animal. They're mixotrophic and gain about 90% of their metabolic inputs from their photosynthetic endosymbiotic algae that they keep within their tissues. They feed off of the photosynthetic outputs and in turn feed the algae from their metabolic outputs. The other 10% of their energy needs come from their ability to hunt zooplankton in the water column. Stony corals specifically are incredibly vital to coral reef biodiversity as they provide the rugosity and little hiding places for all of the little reef fish, as well as the reef predators and everything else that's involved. So the keeping of our stony corals diverse and healthy is incredibly important to our overall oceanic biodiversity. One of the most relevant issues that we see in the media facing coral reefs is their bleaching. So coral bleaching occurs when a coral is stressed and therefore expels those photosynthetic algae from their tissues, allowing you to see through to the bright white skeleton that they have underneath, giving them this bleached appearance. So even though they don't look great, they are not dead at this point. They are able to survive for a short period of time before they must reuptake those algae or they end up basically starving to death, unfortunately. So how does venom tie into this? Coral feed using their nematocysts, which were very nicely introduced in our last presentation. Nematocysts or cnidocytes, which are the overall broad term for the stinging cells that are hosted by cnidarians, come in several different types. Nematocysts are what I will be focusing on as they're most prevalent in stony corals, but you also have spirocysts, casiosomes, and several other varieties. All of these are triggered by mechanical stimulation, again, as we saw in that very lovely video. That was really cool. But they essentially have a hair trigger that exists on the outer capsule of their stinging cell, triggered by mechanical stimulation, which opens the operculum and causes the negative pressure to fire a harpoon-like structure, which sits in coats in the toxins kept within the cell. As we also saw yesterday in our cone snail presentation, cnidarians do possess different toxins targeted for different purposes, deterring predators as well as capturing prey. We know this from the study of anemones, and this is not something that is unique to cnidarians. So with all that in mind, how do we evaluate coral? How do we really look at them? Coral, are they predatory? Are they just waiting around to have a stress event occur and just that's all that they can do? So we start to look at how bleaching impacts corals and their toxins. Toxins are expensive to produce, and these stinging cells are single use. So every time they use them to feed or to protect themselves, they have to replace those in order to continue to do so. So if this toxin production is expensive and metabolically taxing, how does the coral respond when it's in that starving state that bleaching causes? Do they preferentially produce protective or predatory toxins? Do they not impact their toxin production at all? Do they focus on other biological mechanisms to survive? Or do they potentially reduce or halt their toxins without any preference? To start to look at this, we use the model of Stylophora pistulata, which is a well-studied model stony coral species. They have referenced proteome through genome, which allowed us to go through and to do our initial bioinformatics investigation. So we used the genome and proteome of the Stylophora pistulata, as well as fold-seq blast HMMER, the published single-cell transcriptome from Levy et al., and the identification of a script toxin from Brasso et al. Using these, we identified an initial candidate list of 52 initial putative toxin genes and their associated encoded proteins. And to whittle this down to a more manageable list for an initial study, we continued to do downstream vetting, which resulted in a final list of 19 highly putative toxins and their associated encoding genes. These were toxin proteins that contained a signal peptide, allowing them to be secreted, contained cysteine patterns, which are consistent with known toxins, allowing for their proper folding and disulfide bonds, as well as interproscan to identify active domains within these proteins that are occurring in other known toxins of other venomous species. From there, we looked at how coral respond to environmental stressors. We did this using 2 thermal stress transcriptome papers, as well as 1 hypoxic stress transcriptome papers that looked at the whole transcriptome of Stylophora pistulata, allowing us to target our putative toxin genes. To start, we looked at our high temperature thermal stress responses, which resulted in a total of 7 of our 19 putative toxin genes significantly differentiating in response to high thermal stress. In this study, 30 Celsius was their ambient temperatures and 36 their high exposures. Overall, 6 of our putative toxins upregulated and 1 downregulated. That downregulation was seen here in a protein containing a C anemone actinoporin toxin domain with a downregulation from ambient to high temperature exposure of 65%. And our greatest upregulation was seen in a protein containing a dicop domain with a upregulation of 600% from ambient to high exposure. Looking at short-term stress, this was a study that evaluated from 27 ambient to high of 34.5 during a period of several hours where the corals were exposed to these high temperatures. And overall, we saw 11 of our 19 putative toxins significantly differentiate, 5 upregulated, 5 downregulated, and our SHKT metallopeptidase B here upregulated to a 0.32 Celsius exposure, and then downregulated again at that high exposure temperature. This same protein also had our greatest upregulation of 840% between ambient and 32 degrees Celsius exposure. And our greatest downregulation was in another SHKT metallopeptidase containing protein of 98% downregulation from ambient to their highest temperature exposure. So taking in these first couple studies, we start to look at forming our hypotheses about how toxins react. Is this potentially a priority shift that toxins are undergoing during their stress? Are they preferentially upregulating specific toxins and downregulating others? We don't know the uses of these potential toxins yet. However, that gives us something to look at in trying to identify those. This same study also looked at post-recovery. So after coral were set back to their ambient 27 for several hours, there were 5 out of our 19 putative toxins significantly differentiating, with our first protein SHKT metallopeptidase that we looked at continuing to downregulate 95% from those exposed to only ambient to those exposed to a high of 34.5. A third SHKT metallopeptidase, you'll find we found a lot of them were impacted by this, had an upregulation from those exposed to ambient to those exposed to the high temperatures, 580%. This study also looked at how long-term heat stress impacts coral's transcriptome response. And during this long-term stress, which was over a period of several days rather than several hours, we saw 5 out of our 19 significantly differentiated, with 1 upregulating and 4 downregulating. Our greatest downregulation was between those exposed in 32 and 34.5, with a 99% downregulation between those groups. And our greatest upregulation was actually seen in that same second SHKT metallopeptidase as in the short-term study during the same 2 comparisons of ambient to 32. However, this time it only upregulated by 175%. So then we continue to sort of think about what this might mean and look at, is this a breaking point for the corals? Are they not able to cope after a certain period of time? Or potentially, are there other biological mechanisms that protect them from this heat stress that are slower to kick in, and therefore they don't have to prioritize the shift of their toxin productions as much? Again, looking at post-recovery, we see 2 more of our SHKT metallopeptidase containing proteins, with our greatest downregulation of 75% and our greatest upregulation of 465%. Again, 5 out of our 19 were significantly differentiated after the recovery time. 2 out of these 5 were continuing to significantly differentiate from during the active stress response. And the final environmental stressor that we looked at was hypoxic stress. This was comparing non-hypoxic to hypoxic conditions. Overall, all of our putative toxins showed a differential expression at least one time point. You'll see that there's one missing here. It wasn't included in the original transcriptomic study, so unfortunately we could not include it in our analysis. Overall, 6 upregulated and 12 downregulated. And at the initial 12-hour point, we had 4 upregulating and 4 downregulating. This was also where we saw our greatest downregulation for this study of 95% in that sea anemone actinoperant toxin domain-containing protein. After this time point, 4 of our putative toxins returned to a non-significant change in expression level and did not significantly differentiate later on in the study. At week 1, we had 3 upregulating and 6 downregulating. And week 2, we only had 2 upregulating and 8 downregulating. This was also our greatest upregulation that we saw in a PLA2 domain-containing protein with 206% upregulation. So what do we take away from this? What do we start to look at? These are all putative toxins. How are we moving along with this? So we start by looking at what does this mean? We seem to be able to show that there is an increased adaptability in low stress, short-term stressors. There's a greater increase in the number of putative toxins that we're expressing, as well as higher stress responses that we're observing. As you can see, just a summary, 5 out of our 19 for long-term stress, 7 out of our 19 for high heat stress, and 11 out of 19 for our short-term heat stress. This wasn't exactly the pattern that was followed for our hypoxic stress. However, we did still see a shift in the expression over time. So moving forward, again, these are all putative toxins. Are we looking at adaptation? Are we looking at tapping out? We still can't really answer those questions with this data. So then we move into what will be the brunt of my PhD research. We'll start by extracting whole intact nematocysts from healthy Stylophora pistulata specimens and using mass spectrometry attempts to characterize and identify the toxin arsenal contained within these stony corals. At the moment, we don't know, other than that one script toxin for certain, what corals possess in their venoms. What proteins or toxins are they carrying? These are my corals that I have currently at the university. They're doing very well. So with this identification and confirmation of the toxin arsenal, we then move into our bleaching experiments. We're going to look specifically at how the bleached state compares to the healthy state, rather than, as we were looking at in the putative toxin identification, how the actual stressors impact them. Simply in those 2 states, what is the difference that we're seeing? We'll do this by causing the expulsion of algae from our Stylophora specimens and placing them back into an ambient, comfortable environment, however, a sterile one where they cannot reuptake these algae. We'll look at, in that healthy versus a bleached state, how is there a potential difference in the nematocyst content? Is there a difference in which toxins are produced? Is there a difference in the level of toxins that are produced? And so forth. Additionally, we will look at overall nematocyst density. Are they replacing these stinging cells at the same rate while they're in a bleached state as they are in their healthy state? And then moving forward, we'll continue with this research, and hopefully there'll be some really nice implications at the end, starting with just how stony coral respond to the stress of their environment. This will give us better information about stony coral biology, as well as hopefully a better understanding of the timeline that they face when these bleaching events occur. Right now, we work by degree heating weeks to look at how impactful a bleaching event will be and what the mortality might be. Understanding a better idea of how coral are surviving for that short period of time will hopefully give us a better idea as to that timeline itself. Additionally, we're going to hopefully use these venoms to examine protein or venom evolution. As stony corals are quite an ancient phylum, or part of quite an ancient phylum, we're hoping to use this to continue to examine gene duplication, neo and subfunctionalization. There was recently research published looking at reverse recruitment that's been found in sea anemones. So looking at if that's occurring in stony corals as well as assessing the rapid evolution of venoms versus more conserved venoms, as you see in anemones have very rapid evolution, which we, in our initial sort of examinations, does not seem to be reflected in our stony corals. And of course, as we've seen from many lovely presentations this week, there are biomedical implications of any novel toxin discoveries, and while that will not be something that I am personally doing, I hope to be able to put forth some very nice ground-laying venoms for those who are involved in the biomedical communities. Just a quick thank you to my supervisors who have been introducing me to venoms and all of this analysis, and I'm happy to take any questions.